Okay. Good afternoon, everyone. So we're the MRS fund group. We're trying to link neurochemistry to brain structure and function. Uh, Quick side note, neither me nor Frankur here have any knowledge on neurochemistry, so we'll try to do our best. With that note, let's get started. So, okay. So big picture overview, what MRS is and why it might, how we might go about this. So MRS basically captures neurochemical composition of underlying metabolites like glutamate, GABAergic, acetyl and a bunch more. So the idea is to see if those which MRS roughly captures can be related, the underlying neurochemical signature can be linked to structural MRI, either through volumetric information surface or etc., or resting state functional connectivity, or even task fMRI. So we are using so the objectives for this work project was to utilize resources in the HCP aging database to obtain pre-processed information, pre-processed task data, as well as resting state, and in addition, take advantage of the multimodal imaging derived phenotypes or the IDPs, which they already have present there. The second objective was, after we did that, to see how these may be related through MRS and features obtained from task-based fMRI scans. Finally, we hope to see if we can fit some random models to compare the impact of longitudinal scans over baseline, because the ABC dataset happens to have some longitudinal scans as well. So we wanted to see if longitudinal scans will give us a better predictor over the baseline scans. So a brief introduction of ABC. So this is a pretty rich dataset, which has a lot of participants, like 1390 participants with almost 2,800 visits. However, we are interested in MRS, so that is a subset of 282 participants, still pretty big. It has high resolution T1, T2, resting state, task fMRI, diffusion, ASL, and their derivatives, derived parameters are the IDPs. So we thought of taking advantage of this to see how MRS and structure and function can be linked together. Some more interesting things about ABC. So the tasks they have are, there are three tasks they do, have the participants do. So one is the visual-motor task, which acts as a localizer for visual and motor networks. Then they have a carrot task, which is a modified go-no-go task, which includes rewards. So it kind of identifies reward control interactions. And then they have a face naming task, which acts as a task for memory encoding and retrieval. Sorry. Before we head into some analysis and steps, I wanted to show you visualization of one sample participant. So thanks to Noah and Kelly to painfully help us download all this. So we had to put it. Basically, this shows the surface plots of 360 parcels according to Glacier's MAP Atlas on the left, and on the right is a subset of the functional connectivity derived from the same participant. So far so good, right? We see something and which seems kind of relevant, but yeah. So now we are thinking, how do we go from brain parcels, which are 360, a lot, multiplied by 282? Again, it just quadruple. I mean, the number is just is so big, to connecting, making sense of functional networks, which are really, I mean, like 7 or so. So first step is to obviously divide the three parcels into networks or components. We didn't use any ICS, so we just used HCP utils package. Thanks to people who introduced this to us during this 2 weeks. So that gave us networks in YAO 7, 17, and Cole antivariate brain-wide parcellation networks. So we'll be using those to get within and between network FC features. And we'll try to use linear regression for baseline and linear mixed effects models or GE models for longitudinal analysis. On the right is, yeah, this looks as unfamiliar to you guys as to me. Like I mentioned, I'm not from biochemistry. So these are the list of 15 or so metabolites which HCP aging provides. So some of the ones may seem familiar to you, like glutamate, uh, total choline, and aspartate, and glucose at the bottom, and others are slightly less familiar. But because this is more exploratory, so I try to not single out one or two molecules, but do like a omnibus test against everything. Uh, so this is the associations of functional connectivity to MRS. So for this, since we bothered Kelly to painfully help us download all this, so we couldn't let it go waste. So these are results from 42 subjects, unique subjects we derived. She helped us download. Total was about 58. Some of them were duplicates or had longitudinal visits. We didn't use that for this particular analysis, and try to link use within and between network features to across these three different atlases to see how well we can predict. So the ones on the left, as you can see, most of them have R squared in the negative. They pretty much did nothing. It's not a surprise in 42. And running elastic net with 42 with there was training on 34 and held out sample was 8. So pretty small numbers to be doing all this, but there were a couple of interesting things like for MINS and NAA, you see limbic associations to salience and deformant network, which kind of is interesting. Though the R squares are still in the 0.1 range, but there's something. There's some signature for future. So next thing we did was to take advantage of the IDPs. Remember, we had although the sample size for the working with raw data around images was only 42, but there was no such restriction for working with image-derived phenotypes, which we got the whole breadth of the sample, the 282. So from there were, I think, 236 participants who had unique data, so or rather the baseline data. So I used FDI-corrected linear regression analysis to see that macromolecules like and glucose were negatively correlated with structural features, others were positive. And gray matter volume as a feature within these networks, visual, somatomotor, and protoparietal was also negatively associated with glucose concentration at baseline. Sorry? Oh, wow. All right, I have to really skip through all this. So there was a bunch of analysis against exploratory. This is not R squared, this is standardized betas. So everything, these are all FDR corrected. So they kind of pass the threshold, but what they mean or how it's well beyond the scope for this short presentation. I'll rush through. So I took advantage of the longitudinal data to run some GE models. As you can see, there are a lot of so to summarize this in a couple of seconds or a minute, most of the metabolites are found in the baseline were also revealed here, but much stronger. We showed that the longitudinal analysis really had one up over the baseline analysis. So I also looked at the functional IDPs. So for the functional IDPs, only the resting state functional connectivity matrices could give me something beyond after FDR correction. The task-based FCs didn't predict anything. So there were only three candidates here, total choline and NAA could, and also they were between network predictions. So it's a little hard to interpret, but they were positive and passed the FDR threshold. Again, these are longitudinal associations. I will not go into detail, but I rank them through the class condition on the left, task condition, and yeah, you can see some of most of them are showing betas on the right, which is kind of meaningful. To summarize, structural MRIs phenotypes are kind of more stronger and more stable than the functional effects. Pooling visits or longitudinal models give substantially increase the power. So, and NA was the most consistently implicated metabolite across all the analysis we have done so far. In future, we hope that we will be able to pre-process the data not just in 42, but all the 232 samples. And then again, if not relying on their pre-processing, but by using own custom pre-processing, we can ask more better questions about task specificity. Linking these FCMRS or structure MRS pairs to other clinical outcomes will be more useful. And yeah, sorry, that's all for now. Thank you.
Excellent, thank you. All right, next up, we have hyperpace, and on deck is structural and functional connectivity of face recognition pathways. All right. You lost your slides.
Keep working on it, so. No, it is. I just clicked to this, right? Yes. No, the arrows. All right, hi everyone. It is my pleasure to have gotten to work with these lovely people over the last week to ultimately realize that my brain data was unusable and pivot to working with other brains. So this is Hyperface, which was formerly Heather's brain, and we analyzed the Hyperface data set in their dynamic localizer task. And so as you can tell from that introduction, we had a long and windy road to get to where we were going. We began with my brain, which is there, and then we quickly realized that due to a limited number of runs, and also maybe due to my lab manager deleting the events file for my data, that we could not use my data to do any of the analysis that we wanted to do, but we could look at it and it is pretty. And so we ultimately looked for another data set that would allow us to answer kind of similar questions to ones we've been thinking about. And so one data set that came to mind was the Grand Budapest Hotel data set, which ultimately divides into a bunch of other smaller data sets that have participants looking at different kinds of stimuli categories in order to look at the neural representations of those categories. And that led us to making representational similarity analyses pipelines, which allowed us to make representational dissimilarity matrices, among other exploratory analyses. So out of the ashes of my brain rose like a phoenix, the Hyperface data set, which you can see here. And from that, we were able to create a bunch of different types of pipelines that allowed us to answer both these questions that we wanted and also to get the kind of experience that we were really aiming to achieve through learning to use NiLearn, exploring multivariate approaches, understanding how first-level general linear models can be used as input for other analyses, and then basically just throwing a bunch of different approaches at the data set that we had, or that we now had, so that we could really get that kind of experience, move forward with our own data if and when we have data with event files around. So a little bit of background on Hyperface really fast. It's a naturalistic fMRI data set. The great thing about it is that it uses more naturalistic-based videos rather than just still images. So participants are watching these 4-second videos that are associated with either different emotional faces, or when they're looking at when we're looking at the localizer task, which I'll show you in a second. We're also having participants look at 5 different conditions, so faces, bodies, objects, and scenes and scrambled objects, and so that allows us to really see some really rich comparisons across these stimuli sets. And so our main methodological streams of analyses were first doing a localizer RSA. And so that was just, again, looking at that localizer task that has those 5 conditions and doing a representational similarity analysis on that. And we also looked specifically at the fusiform face area and did analyses using neural RDMs with that data as well. And I'm going to prelude Jillian's wonderful section by saying that we were first working with the animacy hypothesis. And so if you're familiar with RDMs, this makes sense to you. If you're not familiar with RDMs, basically we say things that are human and move, faces and bodies, animate, good, correlated. Things that are not animate, will not be correlated. With that, I'm over to Jillian. Thank you, Heather. All right. So first we'll start with our localizer task. So first we started with just one subject 005, while we were developing our analysis pipeline. So first we fit this standard first level GLM to the localizer data. So here we're looking at faces greater than objects, contrast, which we used kind of like as a sanity check to make sure that the localizer was working as expected. So as you can see by the red arrow, and I think, okay, they showed 2 circles here showing that it did in fact work.
So this is just showing the bilateral face selective activation. So the the anterior FFA regions left and right, and at z equals -14 slice. So this kind of gave us some confidence going into our next analyses. Okay, so just because we, limited time, I'm only showing our group RDM, but our first lesson here was don't overinterpret one person's RDM. So originally we had just looking at one person for subject 005, there was essentially like no evidence for representational structure that we expected, following the animacy hypothesis. But once we repeated this analysis across all 21 subjects in the data set, we did see this sort of reliable structure emerge. So our animacy model was significantly captured through this group level structure. And basically, this just goes to show like our original animacy hypothesis wasn't necessarily wrong. It just wasn't detectable in the single participant that we started with. But animacy also wasn't the best fitting explanation among the models that we tested. So this slide here is just showing some of the other models that we looked at. So animacy, space selective, object versus scene, and intact versus scrambled, which is just the scrambled condition compared to everything that was not scrambled. And so although the animacy model did significantly fit the group level neural structure, we want to ask whether it was actually the best explanation for that structure. So as you can see here, the object versus scene did show the strongest correspondence compared to our animacy model. And in contrast, the intact versus scrambled condition was basically null. And so our this leads to the next part, which is looking at faces. So we looked at a more specific question about whether or not face representations within the FFA, whether or not this neural similarity between faces corresponds to people's behavioral judgments of how similar those faces look. So basically, if someone is if people are looking at two different faces as similar, what does the FFA also represent these faces similarly? And so we found that the neurobehavioral correspondence was in fact significantly above 0 in both left and right hemispheres, right on the left and left on the right. And so basically, although these correlations are fairly small, there was reliable correspondence between behavioral and neural similarity across participants, particularly with a larger on the right. But after FDR correction, this was not significant. Okay, next. So next we wanted to basically unpack whether or not this overall neural behavioral correspondence by asking which specific face attributes are contributing to the representational structure in anterior FFA. So we looked at 5 things: gender, age, ethnicity, expression, and head orientation. And basically this is just showing head orientation showed the strongest correspondence with right compared to left anterior FFA and did in fact survive correction. And so main conclusions, basically what I already recapped. So group localizer RDM was a better fit by the object versus scene model than our animacy model. Representational structure was not detectable in our initial single participant, but emerged at the group level. And head orientation showed a stronger correspondence if in the right compared to left anterior FFA. And yeah.
Cool. Okay, in the last minute or so, I'm going to go through a couple tangents we tried to do and one exploratory analysis super quick. So we, in addition to doing all of what Jilly just described for us, we also learned about fMRI prep and practice with that, which was cool. We did do some analysis on head edge brain, but you don't get to see that today. See us if you're interested. We also distinguished animate from inanimate simulees using an SVM, a support vector machine, with whole brain searchlight approach. Also see us later if you're interested. And then we analyzed emotion representations in the hyperface data set. So all the analyses you previously saw were with the localizer data, but using the actual hyperface data set, There were all these different facial expressions. I basically wanted to figure out how the brain actually processes these different expressions. So we wrote code that extracted their brain's activation pattern for each of the 7 emotions and compared them against each other. And basically the most common emotions look largely identical. So you see like happy, neutral, sad, surprise, all triggered these highly similar brain patterns, but fear in particular, oh, you can't see my cursor. Oh, you can. Fear in particular, this green area, were processed completely differently from the rest of the pack. And you might be thinking that looks like an artifact. Well, oh, that's exactly what I just said. It would be an artifact unless perhaps they decided to only have 2 fearful faces in the entire 707 face data set. So the authors stated that they created the clips by sampling publicly available YouTube interview videos to capture faces under naturalistic conditions. So I guess under naturalistic conditions and interviews, it's hard to find all the faces you're looking for. So maybe when you're looking at your data, consider the stimuli at hand. And basically just to further add to, well, yes, it's super imbalanced. Just to add to the results from our RDM, we also ran a correlation of basically showing how distinct a brain pattern was for a given expression and how much it appeared in the data set. And you can see it's super negatively correlated. So yeah, all of that to say, definitely look at your stimuli before running further analyses. And that's all. Thank you.
Okay, next up, we have structure and function of face regions. And on deck is why there's no sense, or why no sense. Sorry.
Hola. We are the team that worked with the working memory task and face recognition pathways and in collaboration with a Zoom person. So let's get on to it. Oh my gosh, wait. Oh yes.
Alright, well, we know that a brain lesion can interrupt our ability to recognize faces. And from data from patients, we know that some white matter tracts are important for recognition, face recognition, because when it's interrupted, you get face blindness, right? So we also know that in the general population, face recognition ability is also diverse. So there are individual differences, and there's a big hypothesis that developmental prosopagnosia or developmental face blindness could be also that individual differences in the general population. Therefore, the Human Connectome project, the HCP data, could be used to track those same connections between face areas to see if they correlate also with the face recognition abilities in the general population. We build a pipeline to do that with 30 subjects, expecting to scale it up to the 1,200. That didn't happen, not for all the data. So we run functional and structural connectivity analysis on the tracks and other areas that we should expect to be correlated with face recognition abilities. So we tried structural pathways and functional connectivity in 2 ways, resting states and task space, when people were looking at faces and making judgments if they've seen the faces before.
So for our dataset, we elected to use the Human Connectome Project dataset. It was a dataset that we're already familiar with, thanks to our work in the workshops earlier last week. And we knew that it contained a working memory task with facial recognition in it, as well as pre-processed functional and structural data. So we elected to take 2 different approaches within the group. We would try traditional correlative analysis between structural measures of the large white matter tracts in the brain and cognitive outcomes. And we would also try a more innovative approach using machine learning models on structural and functional connectomes. So we quickly realized that with the sheer number of edges that would be available in the connectomes, we would have to simplify things somewhat. So we elected to use principal component analysis to do a dimensionality reduction and make it a bit more tractable to work with.
I think saying we were familiarized with the data set is pretty vague because the HPC is pretty big and the safety files, well, they are troubleshooting. So the first challenges we encountered during the analysis of our data, if it was not enough said in the panels from the previous week, well, 80% of our time was used analyzing and watching our data and having to pivot back to the data. And since we obtained like the data from 3 different buckets from the Amazon cloud, well, we did it, but we did it for 30 subjects, and it's going to take a longer time to do 1,200 subjects. So the first thing we did was just look at the runs, and we just had 2 runs, left, right, left for both hemispheres, and we needed to concatenate them to create the template and move from the subject space to our template space. So doing that, we ran out of storage in the GitHub, so we needed to have help, and Noah helped us achieve that so we can move forward with it.
Other issues that we encountered were some pre-calculated resting state functional connectors were already public on the Glacier Atlas, but we found that they kind of merge both hemispheres, losing very, very important details. So we had to pivot into the Schaefer Atlas. So then we proposed of doing the functional and structural connectons based on the Schaefer Atlas all at once, so it can be compared between all of those. And then on NiLearn, you get to actually take care of the NaN values. Importantly, it took us a while to figure those out, but it works so with a mask.
So we also intended on using structural connectivity in our model, and the structural connectomes based on the streamline counts are already available in OpenNeuro data. But in order to more deeply understand the microstructure of these tracks, we wanted to use FA-weighted connectomes. And in order to make the transformation between the FA-weighted and the streamline count, that required the tractography file, which was available in OpenNeuro data. But they also didn't have the FA maps, which we were able to generate, but then we realized there was also no subject space parcellation images, and that was going to take too long for us to generate this week. And so we decided to pivot and look at just mean FA for tracks that are involved in face recognition and the association with working memory tasks.
So yeah, we thought we'd look at associations between track FA for the tracks of our interest and accuracy in the task, in the facial perception task. And so at first we were just looking at the IFOF and the ILF, which were the tracks we were interested in. And we saw that the right for both the ILF and the IFOF were significantly associated with accuracy, which is good. That's what we were expecting. And then we thought we would add some more tracks, like control reference tracks. Okay, cool. Yeah. And so we thought these would be control tracks. Turns out the most significant track was the cortical spinal tract in association with accuracy. So we thought maybe this had something to do with reaction time. So we did the same ANOVAs with reaction time instead of accuracy, and there was no association whatsoever. So we have no idea what's going on with the CST.
Okay, thank you. So we also did the functional connectivity indices. This is an example of one participant. So this route wasn't really successful. We tried several models predicting the performance, the face memory performance from this data, but all negatives are squared. So we understand previous groups that had the same. So up to next one.
So the good news is that we did get our pipeline working. We were able to take in structural and functional connectomes of any size and generate principal components from which we could create data for machine learning algorithms later downstream. We also did bootstrap analysis just to have a look at the components themselves, see what's contributing to the makeup of the components. And it was nice to see that, for example, in the working memory face recognition task, the principal component was primarily being driven by a major hub being located in the ventrolateral prefrontal cortex, which is associated with encoding face identity. So there is indication that the PCA is actually extracting features of interest that we think will be relevant to the performance of subjects.
So we decided to conduct an additional analysis using the PyAFQ outputs by using ridge regression with leave one out cross validation across all 24 tracks to determine whether there was a relationship between individual metrics, track metrics, and phase memory performance. So we were able to conduct this analysis on a data set of 1,030 subjects. And ultimately, the results show significant, there's significant results for both FA and MD with low correlations, but so if we look at the feature rates for both FA and MD, ultimately, based on the R, we understand that, for FA, it only accounts for about 2% of the explained total variance of the data. But it's important to note the inclusion of the ILF, the right ILF track in the top 10 absolute weights for the model.
And with all that said, we can just see that this project lays a groundbreak and groundwork for applying the model to the 1,200 subjects and expecting good results and a future application for developmental prosopagnosia. And thank you all for your attention, and special thanks to Noah, Ariel, Elizabeth, and Kay for all of their help.
All right, next up we have Y neuroscience, and on deck we have the third portion.
Okay, so the arts group, it said, which is my project, will sign up. So that's my project is the Y neuroscience, but there are multiple projects and multiple people.
Yeah.
So.
I think Okay, hi. We're really excited to share a couple of modalities exploring the intersection of neuroscience and art. Yeah, dramatic. And I hope if you've been tired of looking at data, maybe this would be a color. Okay, so we'll start off with Henrik's. So we have a couple: film, interactive, visual, and poetry. So let's just get started with Henrik's.
Yeah, I have.
Okay, sorry.
One thing to say before we.
Yeah.
Oh. Oh. How's the sound? Oh. How do we get the sound connected to the?
It should automatically appear.
No, it's just
It's not that. It's not that. Well, I need to play. It should be playing over soon.
I also have something to say before we start. It's just, it just sounds or seems like the sound is on the Mac, which kind of kills the.
Sorry, I don't know why it's not playing through Zoom.
Okay, wait, sure. So
You might.
Thanks.
You might be able to go to the sound.
Maybe.
Right there.
Try this one.
But I'm honestly not sure.
we'll drop the link.
Yeah, there we are. Yeah. So on Tuesday we decided to make a film. I got Daniel and Lara to go in the scanner, and I got some help from Kelly and John. So thank you so much. I'll start just off with the thought or the question I had when I made this snippet, which was light. Can you turn the light? That is in front. Okay, cool. So my question was just why do I care about the brain? Because it's so cool to sit in code and do stuff all the time, but I got into psychology because I care about something more than just sitting down coding on my own. So that's what I tried to do. So let's see. Can I turn off this? Or it's not? Okay. I've spent a lot of time, so I'll leave it at that.
So for my project, I try to visualize the evolution of neuroscience. I think this is incredibly complex data, and typically the formats which we use to communicate about science is with a PDF file. If you have a dynamic evolution, this is hard because a PDF file does not update on its own. So I don't know really how to present. I'll leave out the sound for now. I used the PubMed database to extract 3.13 million articles in which I looked at all neuroscientific abstracts, and I represented this into a 3D space, a semantic 3D space that large language models use to reason with semantic meaning. So I downloaded 8 atlases in which I tried to represent the evolution of neural entities. So I started off with a basic neural gradient, in which all the incoming papers about these neural entities are mapped onto this space that I transformed from a Cartesian space to a specific template-based space. Then also fungal network. I think a fun fact is that these are the largest neural entities that exist on Earth. Third one is the elegans. So these are ringworms. They are often used in neuroscience. The second one is the Drosophila, so fruit flies. This is an actual brain of Drosophila. Then a rodent, and I chose for the beaver. And the next one is a macaque. This one is often used to model social functioning, which has a lot of generalizability towards humans. The prior to last one is the Homo sapiens. So as you can see, all the incoming papers from, for instance, 2008 are mapped onto this state space. And for me, it's humbling to see that whatever we do is just a small addition to this larger high dimensional meaning of trying to map how the brain works. For instance,
these papers in this specific thematic cluster add information about methods and neurotechnology. Purple information represents information about cognition and language. Do you have the time? Okay, it's not so much. So the idea is just to represent this actual evolution of how we add knowledge to the ontology, to like the unified central area in which we all try to communicate about science. So I think that's it.
Sure. Okay, so as you can see from Henrik's and Stain and Daniel's visualizations, wait, don't read it yet. Yeah, neuroscience research has been going on for a long time, and it probably will, hopefully, for a long time more. And there's a sense of curiosity and devotion that I think we bring to the field, and I hope my poem can also convey that. Don't reach too far ahead. Okay. There's an arched neuron nestled deep in the medial prefrontal cortex, spindly and fibrous, spanning the interstitial fluid. It floats with little zaps of mind coursing through its ends, forever knotted with the axon terminals of the ganglionic layer. Nearly 2 centuries on, the myelin is tight and the hollow of the synapses holds its crisp memory well through the action potential's startling speed. Situated in the midst of evergreen bundles, its dendritic arms is past the upheaval of developmental pruning. The track is robust with voltage gates. A break in its branch sets the path alight. It pulses in sepia-colored dreams. I've come to this place since I was a child, before it was ever cartographed by fMRI. Brought first by my parents, then as a teenager with Raven, whose research on consciousness had us collecting neuron samples from the little zap of mind within. If I could be sure that this neuron fell exactly within Brahman area 32 or your parcellation 7.3, I might lay down here on the smooth, softly charged causeway of its tracks for all that's left of time. The nucleus of this neuron was laid down long before the tissues we know of today ever interweaved, then pulled apart. Hundreds of millions of years have passed since the neuron's heart was laid down as a student of the amygdala. If I knew that I could, I would lay here, awash in plasma, veins of sentience threading thick above, listening to its little zap of mind until the world flooded anew. As a new student in the same world, I would welcome new blanketed layers of being until those hundreds of millions of years passed, and through the pressure and heat of development, I would become the same nucleus buried in the heart of the mind that man has so intensely yearned to map. And one day, after the interstitial fluid of a flooded world recedes, that social affective neuroscientist would find me and put me in an fMRI machine. And perhaps then, released into the magnetic field of the K-space, I would be with Raven, who, having been birthed so many millions of years before, was with me as I lay down to hear that little zap of mine. Thank you. Yeah, that's it.
Amazing, thank you. I think this might be our first art project. All right. Next up is Study Forest, and on deck is NeuroLabel. So Study Forest, please come down.
Can I go first? We thought we were one after. Yeah. Based on the list.
No, you were next, but I mean, if someone else volunteers, better be quick. Come on. Who group you? Large language models. Okay. Yeah. Okay, we're gonna instead have large language models.
My name is Priya.
I'm Gavin.
My name is Sam. So for our project, we wanted to leverage the pre-trained knowledge of large language models to predict clinical phenotypes in a way that you don't need to train any new models. So this will be a speed run, so heads up. One of the reasons we are interested in using large language models is because conventional machine learning algorithms are not scalable in the sense that it's not able to work as a general purpose probability machine that can ingest basically any type of multimodal data to predict any type of clinical phenotype. So one of the potential solutions that I think is interesting to explore is to use large language models, because during training they learn to predict the next token distribution of a previous token set. So it's, for instance, reads a lot of information about the literature where it establishes a relationship between the brain and phenotypes. So for instance, it reads the word hippocampus and it knows that it is related to memory more often than other words. So you can look at the next token distribution as an indication to predict these phenotypes. So for my internship, I created a tool. I won't go over all the details, but the idea is to use a multi-agent setup that uses a self-supervised quality control check to optimize the likelihood that a prediction of a clinical phenotype is correct. If you're interested in it, it's called the Compass engine. So I validate this with UK Biobank, where I needed to create a linguistic representation because large language models use tokens. So what you do is you create a high resolution linguistic representation using an ontology, which is a formalized way of capturing a very complex state space. So the motivation behind our group project is to extend this clinical validation effort with open neural data sets. So we looked at very diverse multimodalities and very diverse clinical phenotypes to predict intelligence, first episode psychosis, and numeracy after stroke. Then our overall methodology is to loop through two loops. So the first one is through data complexity tiers, in which we always started by demographics, then we added clinical variables, and then we added all the brain data, and then the last tier was always that we only looked at the brain information. Then the second loop was through large language model providers to do like a cross-provider interreliability check. Yeah.
Okay, the first data set we have is a stroke patients data set where they collected 108 left hemisphere chronic stroke survivors with a mean age of 61 years old and then a mean aphasia severity of 93 out of 100, but reverse meaning it's just right below normal language ability range. So the patients performed two tasks. One is approximate numeracy, where they compare which cloud has more dots. And then the second one is precise numeracy, where they indicate which digit they just heard. This is an example of subjects with gym mask. The yellow area is the damage area. This is the voxelized correlation with approximate numeracy, and this is with precise numeracy. So the lighter the blue is, the stronger the negative relationship between the damage and numeracy performance is. This is our correlation map of some of the features that includes demographics, clinical assessment, and some brain areas. We ended up dropping some of the very red factors because turns out they are derived from the same math task that numeracy was from. And then we performed some transformations on some of the features and created an ontology to linguistically represent these features. So at the end, these are all the features that we use to fit the large language model. To predict, we fitted large language models. We're using several tiers. We always begin with demographics information, and then second tier we added clinical assessment, including aphasia, and then third tier we added lesion masks. So as you can see, as the tiers goes up, model performance getting better and better. So we found that across tiers, adding lesion files achieves the highest performance of 0.57. And then across providers, Laguna model achieves highest performance of like 0.62.
And different data set that we wanted to test was a first episode psychosis data set we found on Open Neuro. So this had a healthy control group and a cohort of participants who were positive for first episode or ongoing psychosis. They went through an EEG resting state task. There was no task, they just sat there and had their brains measured by the EEG. And our target phenotypes were to see if the model could predict diagnosis status, so control versus FEP, first episode psychosis, or symptom severity for things like anhedonia, hallucinations, etc. We extracted, and here's where I had my notes that I just lost. So hold on one second. Oh, great. That was easy. And we extracted over 800 variables from the EEG data, ranging from spectral power to alpha peak, entropy, self-similarity. We even had some variables inspired by graph theory and, of course, microstates. And here are graphs that we generated comparing the two like diagnostic groups. We're seeing some differences, but not a ton amongst our variables. And we used that to generate an ontology of linguistic representations of these variables. And this is a correlation matrix of our EEG, or sorry, this is our non-EEG data, because we also had demographics, intelligence testing, global functioning, and social functioning from structured clinical interviews. As far as performance, so for predicting whether there was a diagnosis or not, it did extremely poor. This is a confusion matrix. You want to see the darkest squares should be from left to right, like top corner, bottom corner. So this, like 61 down here, being like that dark blue is really bad. It means that it predicted control for 61 people where they actually were positive in psychosis. It did marginally better with the symptoms, but overall not great. And we had a third data set.
Yes, so for the last data set, we tried to predict intelligence using MRI data. So a set of features, so I won't go through all of them, but we use demographics, psychographics, and actual brain data, so MRI data, to predict in a hierarchical fashion the total intelligence scores and fluid intelligence, memory-based metrics, and crystallized intelligence. So the same as the other two data sets, we created a linguistic representation of this. So for the prediction performance, the explained variance is not large, but without training, it does significantly predict the fact that a certain individual has a hierarchy given brain data. So this is like an aggregate statistics map in which you actually see that if you add the brain data, that the performance decreases. I think this is because we used very small large language models. So perhaps already a point of our discussion is that this may have exceeded the context window of this of these models.
Right, so future directions, we can explore more models like general purpose models, or those zero-shot models that don't need training data. And across our project, we have been using AI responsibly, meaning the whole cost for us was only $7.5, and we didn't try to token maximize. And maybe further validation with other data sets are encouraged.
We could also experiment with the actual model structure, so which agents are working on the task and sort of how they iterate. Also, computing feature level explanatory metrics, so trying to figure out which features are driving, are linguistically driving positive predictions. And there's also an issue of potential like temporal leakage because, so for example, the open neuro data set that I used may have been published while the large language model we used was trained. So it could have had previous knowledge of that data set beforehand. Thank you.
Excellent, thank you. All right, next up we have Soy Forest. I see the list was updated while we were giving talks, that's why I was confused. But sorry about that, Soy Forest. And then on deck will be NeuroLabel. You need to share your slides.
Okay.
On Zoom.
So I do share. Oh, I won't be able to see my speaker notes.
Oh, sorry. Yeah, that's
That's fine. Okay. Hi everyone, sorry for my voice, but we are the group that explored the study forest dataset and worked on many different subparts of the dataset. So we'll just jump right in. So the dataset provided annotations for like as people were watching the movie, the annotators annotated whenever there was an emotion present, like all kinds of labels. And what I would point out is that there are like some emotions are more universal and more present, like happiness, fear. But there are other kinds of emotions that I think were probably a bit confusing for people and did not really get many labels like resentment. So it was a bit both imbalanced and sparse. There is also this aspect of synonymous emotions present. So if you see like there is satisfaction and gratification both, so that could be confusing and probably contributed to the annotators kind of being divided in their opinion. There are also information which we later used about what cue they have used to annotate this information. So they have the audio, which just means music, and like other non-speech events, there is face, which means that they had used the face to annotate face or gesture or narrators, whatever they were saying to annotate the images. So yeah, I will highlight that people mostly use face and the verbal or the speech to annotate the images. There are also multiple emotion categories that have no label available in them. And you'll also see that certain labels, for example, in this case, I think this is contempt, which is pretty correlated across other things like resent or hate. So again, they had provided too many categories that contributed for people just being divided in their opinion and the general emotion category not being super informative. So yeah, it's just showing that the face and the speech in the movie was most informative for people to categorize. So yeah, so to summarize, the emotion labels are both imbalanced and sparse, and categories are sometimes highly correlated with each other, and that ended up making the people being split up in opinions. And more basic categories would have probably done better. And yeah, I will highlight that the face and gesture are cues that are present in the visual, and the verbal, audio, and narrator are cues that are present in the audio, because this is going to be important for the next analysis, where we looked at the higher level context and emotion processing in one participant that watched the entire audiovisual Forrest Gump movie. So I ran a banded ridge with low-level visual and auditory features, and the emotion labels alongside what audiovisual cues were used to categorize emotions, which I just showed in the last slide. And the banded ridge is basically this nice kind of ridge regression where you have different parameters, different regularization parameters for different groups of features, because they will vary a lot in like scale, range, length, etc. So the basic pre-processing on already fairly preprocessed data within the subject native space, I did out with NiLearn. There was just Z-scoring in everyone, detrending, and the using the confounds parameter neatly to account for the motion correction. So yeah, we fit on 7 runs and then like tested remaining data, did some cross-flow validation with 4 runs and only 1 subject for time. Yeah, the results are reported on held up data. So yeah, so this is for low level visual, and we very nice to actually see the occipital pole being highly predicted by the low level visual beyond other feature groups. So they're already accounted for, and the banded ridge lets you do that. Then the R squared explained by the low level auditory features. Again, this is around the Heschel's gyrus being highly predicted by the low level auditory features. And then we have some interesting thing for like the video cues. So the R squared explained the video cues, which was, again, I'll remind that the face and gesture movement that people use to categorize emotions, those were pretty good predictors around the occipital parietal area. So that was pretty neat. And finally, the audio cues are also around the temporal lobe. The audio cues were
basically the indication of speech or the narrator or the music. So what I will highlight is that these are pretty good scores, R-squared and held out data, given the information used to feed the encoding. They're like this is not like the majority what we'll explain would be like the video itself and like the speech itself. Like you can get up to like 40% in a lot of areas with the right. So I think that this probably indicates we're in the right direction. I did not actually, unfortunately, get much from their emotion. Like it didn't really explain much beyond the other fun things. So this is like a winner takes all thing. But I'll just hand it to Drew.
Yeah, I just plan to do the similar analysis, but with the focus on the primary visual cortex, but I failed at the very first step, which is the localizing the V1 because I had trouble accessing the data and aligning, different spaces. There are like multiple MRI spaces in this data set. And I just pivot, to figure out those alignment and create some useless figures. And yeah, this is the V1 I figured out, and then apparently outside of the brain, and yeah, and this is the retinotopy map of the V1, and yeah, hope it is correct. Yeah, and you had to expert.
Yeah, that's.
Hi,
can you hear me?
Yes.
Okay. Okay. So what I did was to compare speech versus non-speech vocalizations. So these were my conditions in study forest dataset, and just to find which brains are being activated during these two conditions. The dataset that I used was the German audio description of Forrest Gump. So only the participants were only listening the movie and they didn't watch it. This was a 7 Tesla fMRI with a repetition time of 2 seconds. I did a preliminary analysis of one participant with 4 runs because the whole movie was 8 runs, fMRI runs, and I used half of them due to time and RAM limitations. The speech annotation provides onset and duration of speech words and non-speech vocalizations. And I just exclude the 3 vocalizations because they were overlapping with the speech words. So can someone change the slide? So the dataset was kind of imbalanced though. Can you run a bit back? To the first? Okay, so as you can see in the plot, the datasets were kind of imbalanced. So we have around 8,000 words, and the non-vocalization condition were around 100. So next slide, please. So I got some exploratory uncorrected maps. I used the provided preprocessed nonlinearly aligned bolt images, and I create a common for an intersection mask. And I did the first level GLM separately for each run. And motion parameters include destinations and regressors. So this is the uncorrected map that I got. Next slide. And this one is the uncorrected map for the non-speech condition. The 4 runs combined, and I use a precision weight fixed effects. And then I ran, next slide, an RDF analysis in order to see which is the statistical significant regions that are activated. So I ran a 2-sided voxel-wise FDR correction with a threshold of 4.6 to 1. So I have conclude that 44 voxel cluster is the statistical significant. Effect the direction was negative in all 4 analyzed runs, and only 1 isolated voxel survived for the speech. So what we see here is 4 non-speech vocalizations, and the cluster lies within a broader right temporoparietal region previously implicated in affective processing. So these are kind of preliminary results because we need more subjects and the whole movie. But this was what I concluded from this analysis. Thanks.
I also did some preclinical
results on the emotion classification or decoding. And firstly, I use the agent large language model to classify the emotion of the movie over time. It's a two-hour movie based on the annotation of the day site. And to make sure the emotion coding makes sense, I try if it's possible I tried the relationship between different subjects when they see the movie. And actually for the interview Pareto, it makes sense. So I use the Voxel on that to do the emotion decoding, and the result isn't very good, but makes kind of sense. Thank you.
I did
12 seconds.
I did the most standard GLM analysis you can imagine, but I didn't learn that part. And then there's my design matrices, and here's body, face, house, object, scene, scramble.
Also, there's some type of classification analysis where you took like a functionally defined region of interest like he did. I didn't get to this point as maybe this slide spoiled, but I just put it out there because this is also the kind of thing you can do with this type of rich data set. All right, thank you.
Thank you, excellent, nice job. Wrapping these up. All right.
Oh, wait, that's something.
Yeah. Okay, we now have NeuroLabel, and on deck is aperiodic signatures of hormonal contraceptive use. So,
Sorry, I can't share the screen yet.
oh, you're still sharing, Krishpita, did you? Or just quit Zoom, that probably works.
Thank you.
Okay.
All right, while we're getting set up, I'll just begin. Hello, everyone. I'm Amisha, and this is our group, NeuroLabel, formerly known as PyParcel, which we had to change because PyParcel was taken on PyPath. All right, so NeuroLabel began as an effort to make neurosynth parcellations more interpretable. And thanks to the effort of the entire team and also the folks on Zoom, we went beyond our goal and were able to position this package as a more generalized interpretation toolkit that can work with any parcellation, atlas, and even a statistical brain map. All right. And we'll begin by telling you a little bit about why parcellations are useful, the problem we're seeking to solve here, and how our package can help.
All right, so to start, why are parcellations useful and what are parcellations in general? So as we know, and as we've seen throughout this process, the brain and neuroimaging produces a lot of data. The brain consists of many thousands of voxels, and this can be hard to interpret meaningfully just due to the amount of data. Parcellations and breaking the brain down into parcels can help with dimensionality reduction and are more computationally efficient in looking at these regions or parcels that can be determined by anatomical or functional divisions of the brain. And what is neurosynth? So neurosynth is
Sorry.
Okay. Neurosynth is derived from a meta-analysis of task data for almost 15,000 studies using patterns of coactivation to produce this parcellation. And based on this meta-analysis, it also offers a term annotation, so it looks through the article abstracts and extracts out common terms that are mapped onto these different parcels. And neurosynth offers 3 different parcellations, so dividing the cortex into 50, 100, or 200 parcels as pictured here.
So even though neuroscience can be really useful in some contexts, it also has a couple of problems. First of all, the parcel numbers on their own are not interpretable at all. Also, reading in brain maps requires substantial expertise, which some labs might not have, especially if this is a new method for you. In addition, the commonly used centroid lookup alone can misrepresent large, irregular, bilateral, or heterogeneous parcels. And then finally, a lot of the anatomical and functional interpretation of brain maps typically requires manually using your own tools and also customizing your own code. So it can be both time consuming and difficult if you don't have the experience. So, what is NeuroLabel? So it's essentially a package that we developed that provides a unified workflow for Atlas-based anatomical annotation, as well as interactive visualization and functional decoding. So individuals can input their data and they can annotate anatomical regions for certain regions of interest using a specific reference atlas. They can also perform functional decoding of their regions of interest to identify associated cognitive processes and also interactively explore their brain maps. So Atlas overlap is one of the core functions in NeuroLabel functions. By giving it a labeled atlas and a target parcel in another, likely unlabeled atlas, you can get a data frame of how that target parcel overlaps with the labeled parcellation, ordered by the number of voxels in the intersection. So here we gave it parcel 19 in the NeuroSynth 100 atlas and asked how that parcel overlapped with the Harvard-Oxford atlas. And we see that it mostly overlaps with PCC, but includes other pieces of nearby regions. And if we want to visualize this breakdown, we can also use the plot and nat function, cool, which makes a bar plot of the regions with the most overlap. On the previous slide, we showed Harvard-Oxford, but you can give it any labeled atlas, including functional atlases like Shaper 400 on the right. All right, and this is my personal favorite part of the package, which allows you to decode cognitive processes most strongly associated with an ROI. And this can be a parcel within a parcellation, a set of boxes that you found that were significant in an analysis. And essentially what the code function does is it wraps around Naiman's ROI association decoder and performs image to text functional decoding. That is, it identifies terms whose meta-analytic co-activation patterns are most strongly associated with the ROI that you just gave it. And it gives you association scores in the form of correlations. And you are also able to very neatly plot the output of this decoder using plot decoder as pictured right here. Next up, we were able to go one step ahead and build an interactive parcellation viewer, which allows you to interact and explore all the components of a parcellation. So over here, you can see that we are visualizing all the parcels within a parcellation. I believe this is Shaper. And on the right, you'll see functional and anatomical information when you input a parcel number. So for example, over here, I entered parcel 7, and I'm able to see the number of voxels that are a part of this parcel, and its functional and anatomical information. And Open Interactive is a button that's up on the right, and clicking it also gives you NiLearn's interactive viewer, where you're able to plot the brain around and see where this parcel lives. And I think this is a powerful tool also for education and being able to get comfortable identifying where certain regions are in the brain and what they do.
Yeah. Okay. So far we discussed about how NeuroLabel uses these parcel inputs as the input to generate the various anatomical and the functional descriptions. So apart from that, this is an additional feature called statistical map summarizer. I would say basically this is a second entry point to NeuroLabel. So this, apart from accepting parcel numbers as inputs, it would also accept the statistical maps. So these statistical maps could be like brain maps from any of your analysis, say group analysis, GLM, or any thresholding analysis. So these brain maps could be like containing p-values, t-values, or your thresholded binary results. So what the summarizer does is it analyzes the maps and extracts the significant boxes based upon the user-specific threshold, which you might have used for your GLM analysis or the group analysis. And then these significant boxes would be mapped onto your required parcellation, say NeuroSynth or any parcellation of your choice. And then these parcels are returned as parcel IDs, which would be passed again to the NeuroLabel to do the entire analysis. Yeah, and this is like one example output from the summarizer where you can see the summary of the parcels. Yeah, and then run workflow. So in order to make the package easier to use for the users, So this is like a single point of entry. It goes all the way from brain maps or single parcels to the whole anatomical and the functional interpretation. Like you have a single workflow function which takes parcel inputs, IDs as inputs, or a statistical map. And it goes all the way from extracting your parcels and then giving the anatomical and the functional descriptions, as well as along with the relevant plots that's been called from the various functions. So it's a single pipeline. Yeah, and this is the output that's shown here for a parcel number, example parcel number of 11.
Okay, so what we learned is to collaborate, especially on GitHub, and to not overwrite something that someone was just working on. We also learned, I think, to eyeball our data, because we noticed that our first Centroid approach was not working. And I think if we wouldn't have eyeballed it, we wouldn't have noticed. And yeah. And also like maybe a sense of agency that when we run into a problem like this, that we usually I would do a workaround for myself, but I would have never thought of like creating a package for everyone. So this is great. Yeah. Oh, and also we have a little Easter egg. So when you can call Jimothy and look at him when you have too much analysis with your code. Sorry, too much analysis. And the brain is fried. Look at Timothy. Yeah.
Yeah, so we currently have everything up on our GitHub repo and hoping to get this soon on PyPy once we do a bit more testing. But if anyone has any insights or any thoughts or contributions to the project, please let us know. Big thanks to NeuroHackademy, all the organizers. Big thanks to Elizabeth for helping us along the way. And keep an eye out for next week. There's some March. Thank you so much.
There's any large entity make appearance. All right. The mystery up next.
Yeah. Here we go. Okay.
Great. So we have the exact same color scheme, which I'm really excited about. Our project was aperiodic signatures. Aperiodic signatures of hormonal contraception using in resting state EEG. Hormonal contraception is one of the most widely used medications on Earth. 874 million women use modern contraception worldwide. Over 20% of reproductive age women take the pill in 27 countries, and 50% of global contraception prevalence among just global, prevalence. That all being said, hundreds of millions of women alter their hormonal profile daily, yet the effects on the excitability remain poorly characterized.
Awesome. So getting in a little bit into how menstruation as well as hormonal contraception affects the excitability of the brain. There are kind of two main players. So estrogen is an excitatory signal. It boosts glutamate and NMDA. And progesterone is more inhibitory in nature as it modulates the GABA-A receptors. And so across the natural menstrual cycle, we see the balance between these two sex hormones rising and falling. However, when women take contraceptives, that natural rhythm is suppressed and it's replaced with a steady exogenous profile of hormones. So how can we measure excitability in the brain? So one of the ways that we can do this is using the 1/F aperiodic slope, which I'll go into a little more detail in just a second about. But this value indexes the EI balance of the brain, where inhibition is going to steepen this slope and excitation is going to flatten it. So thinking about these sex hormones, estrogen dominant phases or medication should flatten this exponent, and progesterone dominant should steepen it. In a most recent investigation, they were not able to find a link between hormonal status and the aperiodic slope or other periodic measures like alpha power or peak frequency. And so in our project, we wanted to do a confound-controlled multivariate decoding of distributed EEG features with combined spectral and temporal analyses. With the overall question, is contraceptive status decodable from the brain at all? And does hormonal contraceptive usage induce changes in the aperiodic neural dynamics? All right, so getting to our methods a little bit. We were working with data from Open Neuro. We have 69 women who underwent EEG, resting state, both eyes open and eyes closed. They also completed a lot of questionnaires. Those that are most relevant to our analysis are the cycle and contraceptive information. And I've listed some of our key variables here. So like cycle phase, current or past use of hormonal birth control, and the birth control type. So here, just demographic graphs. So you can see we have women roughly between the ages of 20 and 40. We have a nice spread across the 3 phases of the menstrual cycle, and a good spread across current, past, or never using hormonal contraceptives. And so now getting into the spectral features, I'll talk a bit about how we did the spectral analysis, and then we'll get into the temporal aperiodic analysis in a bit. So for this, essentially to calculate the aperiodic parameters, you can take the EEG data raw that's recorded from the scalp, and then perform some really fancy math, a Fourier transform, and then you get this in the spectral domain. In the graph you can see on your right, and that essentially gets our signal into a different domain where we have frequency along our x-axis and power along the y-axis. And so periodic peaks are going to sit on top of this spectra. So you can see we've highlighted the alpha oscillation, bless you. And yeah, and the aperiodic component, what we can do to calculate that, we can fit a model. So we're using SpecParam, which is a tool that was developed in the Wojtek lab, which is Nicole and my lab. And so we'll fit a model to this power spectra, and then you can extract a couple of parameters that are going to quantify that underlying aperiodic structure. So we're going to be looking at the aperiodic offset and exponent. Awesome. So first we did a confound control just to make sure that none of the other variables that we would be inputting into our classifier would reliably classify the subjects into the group of using versus not using hormonal contraceptives. As you can see, the confounds don't separate the groups. So the confounds only classifier decoded hormonal contraceptive use at roughly chance. Now getting into our overall model, the finalized model, what we did here, I'm just re-showing you the confounds only model. We then ran a model only including those EEG parameters, so the aperiodic parameters in resting state, eyes open and eyes closed, which boosted up that performance a bit. And then our final model included all of those EEG parameters and residualized confounds, which are listed.
Cool. So I'll just talk a little bit about the temporal analysis we did. So just kind of complementary to the spectral offset and exponent that we looked at in the aperiodic signal. We also quantified the neural time scale, or basically the time that the signal takes to decay past, well, I guess fully decay, where it's no longer correlated with itself at all in a previous lagged version. And this is thought to basically index the temporal memory of a signal, or the integration window that allows it to remain stable over time. Cool. Yeah, so this is a complementary metric to the aperiodic slope because it's basically just the time domain metric of the same EI balance. And so a shorter autocorrelation window is driven by faster dynamics or more excitable, rapidly changing states, whereas a longer autocorrelation window is more slow, stable dynamics, driven more by inhibition. And so our hypothesis was that estrogen, which, as we talked about, is more of an excitatory signal, would have a shorter, or would produce a shorter time scale with a flatter aperiodic slope. And contrary, progesterone, which is more inhibitory, would produce a longer autocorrelation window and a steeper slope. Cool. So what we found here is that hormonal contraceptive users do have shorter global timescales across the cortex compared to non-contraceptive users, reflecting more transient excitatory dynamics, or perhaps a lack of inhibition. And inversely, we were able to, or the strongest predictor of the duration of timescales was hormonal contraceptive status, as in whether somebody was a user or non-user, and a predictor of how short their timescales would be. And then I'll be quick here at the end, but yeah, so then we looked at the different phases across the menstrual cycle. And as you can see here on the left with these non-hormonal contraceptive users, so naturally cycling women, there's this kind of clear progression across the cycle where phase-dependent kind of duration and variability profiles seem to kind of consistently grow as those hormone levels fluctuate. And versus in hormonal contraceptive users, we no longer see that kind of natural progression across the cycle. And instead, they all kind of trend towards this more just stable state across the cycle. Cool. So just in conclusion, we found that current hormonal contraceptive use is decodable from resting state aperiodic EEG activity. We also found that hormones modulate the EI balance and reshape neural timescales, at least using the autocorrelation 0 measure. And so we saw globally shorter timescales in hormonal contraceptive users due to estrogen and excitatory activity, and longer timescales in the luteal phase, driven by inhibitory dynamics. And so our overall kind of takeaway is that the aperiodic 1/F signal carries real information about hormonal state, and that fluctuates across the menstrual cycle, which indicates a promising understudied marker that is worth further pursuing. Thank you.